and thanks for tuning in to BEaTS Research Radio, a podcast and YouTube channel that aims to disseminate science and research to the community. My name is Nicole Chu, an undergraduate student from the University of Ottawa. In today's episode, we are honored to be joined by Dr. Filipa Simões, a principal investigator and British Heart Foundation Intermediate Basic Science Fellow at the Institute of Developmental and Regenerative Medicine in the University of Oxford. Her research aims to better understand cardioimmunogenomics and how immune cells can interact and contribute to the heart's regenerative response after a heart attack. Thank you so much for joining us today, Dr. Simões.
Thank you, Nicole. Pleasure. Thank you for having me.
So to begin our conversation, I would first love to learn more about your career journey, which led you down the path you are presently on today. Do you mind sharing with us your journey?
Of course. So I'm originally from Portugal, and that's where I grew up. And I went to, I did my undergrad studies in Lisbon, and with a bit of, with a period also in Paris as an Erasmus student. And that was my first experience living abroad, and I absolutely loved it. So I was quite adamant when I went back to Lisbon to finish my undergrad that I would really like to have another different experience abroad. So then when I finished my undergrad studies, I applied for this PhD program, still back in Portugal, that I think was an incredible opportunity because for the first year, you have every week a different subject that you're taught. Basically, they would fly in experts in the field, and I would have, for example, this week I would be learning about crystallography. Next week, I'll be learning about microbiology or developmental biology. So it was really a privilege to be exposed to such different variety of subjects. And by the end of that year, they would give you the incredible, again, opportunity to fund you to go anywhere in the world. And you pick a lab of your choice to do your PhD studies, the research itself. And that's how I ended up coming to Oxford. And I joined the lab of Professor Roger Patient at the Wetherell Institute of Molecular Medicine. And that was really a really exciting and fun time. I was very interested in developmental biology and how, you know, in an embryo, how do cells know what to do? And in particular in the cardiac field, my project was very much related to understanding how a cardiac cell decides to be a cardiac cell over a blood cell. So which programs and which genes are actually in place during very early development to set up this cardiac program. And then when I finished my PhD studies with Roger, I started, still very much with a developmental biology background, I started to be interested in a regenerative perspective and how we could understand how the heart repairs itself more in an adult kind of, from an adult perspective. And therefore I contacted Professor Paul Riley. At the time he was in London at UCL, trying to understand if there would be any interest in us working together to look at how the heart can heal itself after injury, still in the zebrafish. So my PhD studies were done in the zebrafish as a model, and I want to explore this now during my postdoctoral studies. So Paul was very open to this, and he had a long interest in this epicardial layer, which is an outer layer that wraps the heart, and is very important during heart development, but also after injury. And during my postdoctoral studies, I was looking at how programs can be in place and reactivated after an injury event to help the heart to regenerate in the zebrafish. And then after this, I was awarded a transitional fellowship that allowed me to work closely with Professor Tatiana Suka-Spengler, which is an absolute expert in more genomics and gene regulatory aspects of how this is the way she looks at developmental biology, very much with a more genomics perspective. And that gave me the opportunity to learn and adapt a lot of the technologies that she uses to address my own research questions. And that allowed me now to really have that as I started my own independent lab.
Oh, that's so unique to hear how you got your first exposure from research through those rotational programs. And as you mentioned, in your postdoctoral research in Dr. Paul Riley's laboratory, you have lots of experience investigating heart regeneration in zebrafish. Why are zebrafish such good animal models to research heart regeneration?
Yes, thank you for that question. Especially these days, there's been quite a lot of push to do human-centric work, which is obviously incredibly important, and we can understand why. But I think it's also, from my perspective, to focus on organisms that are a bit of an exception. And zebrafish is that exception in the sense that they are champions of regeneration in a way. So not only if you injure the heart can regenerate, but many other organs the zebrafish can regenerate, which obviously contrasts with what happens in adult mammals. So if humans or an adult mice get their heart injured, it cannot really regenerate. It will repair it, but will not get new blood vessels, the new cardiac muscle cells formed. While the zebrafish can, in fact, do this. If you injure the heart of the zebrafish very quickly, within 1 month, you will have all these new cell types generated. So de novo generation of these cell types, which are exact same cell types that you would need in a human setting or in a mammalian setting to replenish the injured, to help healing the injured heart. And because we are if we think about the percentage of similarity in protein coding genes between humans and zebrafish, it's around 70%. So there's a lot of similarity that we could learn, and how these programs are set up in the zebrafish and how we could draw parallels to the mammalian setting. But I think it's also important to focus on the differences, right? Because these are quite, and there are other organisms like the zebrafish that have this incredible capacity to regenerate still today. So evolution gave us almost this present. And why would we waste this opportunity to focus on these animals and to really look on a bit closer? Why are they different? What are they making that is so different from what we do? And just to learn from an evolutionary perspective, I think it's quite an interesting way of looking at the problem.
I really like that perspective. And sort of diving into more specifics of your research, in your paper published in Nature Communications, your team discovered how macrophages directly contribute collagen to scar formation. Can you give us a brief overview of the results from your study?
Sure. So if we zoom out a little bit, and go, we've been discussing a little bit about this concept. So if you or I are unfortunate to have a heart attack, we'll lose billions of cardiac muscle cells, and we cannot regenerate these cells. We cannot get new cells formed. What we have as a default mechanism of repair is formation of a fibrotic scar. And this fibrotic scar is necessary to start with because it holds the ventricle in place, so the ventricle doesn't rupture. But what happens with time is that because this scar is very stiff, it will really impair the heart to pump efficiently blood through the body. And patients that survive a heart attack then most of the time will develop a heart failure. So just by saying this, I think it's quite obvious how important it is to understand how this scar is formed in the first place. And if, to understand its composition and its nature. And if we do that, then perhaps we'll be better suited to modulate the nature of this scar. And rather than having a permanent scar formed after injury, ideally what we could aim to do is to make it transient in nature and be able to remove this scar after a bit of time after the repairing is happening. And this is exactly what actually the zebrafish does. But what me and my colleagues found in Paul Riley's lab with this work that you mentioned is that macrophages, which are immune cells that are very important to fight infection in our body, and they're actually important players and very much first responders after injury in many other systems, we found that they are actually playing an active and direct role in setting up this scar. So at the transcriptional level, we found that these macrophages were both in mouse and in zebrafish, so in a non-regenerative and in a regenerative environment. We found that they were upregulating a lot of the components that form this scar, in particular collagens. And if we think about the scar, it has to be deposited at the extracellular level. So we also found with our work that indeed not only the macrophage was producing these internally as transcripts and as protein, but in fact was able to export these proteins to the extracellular level and actually deposited in this scar. So what is quite important, I think, about this work is that we're now placing macrophages as a new important player in setting up this scar, and therefore we can put this cell type as a potential new therapeutic target to modulate the making of this scar, if you think about moving forward.
Oh, I see. So your team really focuses on this interrelationship between our immune system and the cardiovascular system. So recently you have been also awarded the British Heart Foundation Research Fellowship to pursue this project investigating programming of macrophage function in the regenerating heart. So can you explain to us the main goals of this research?
Of course. So I'm very excited about this idea of programming cells and how we can make a cell do one thing versus the other. And this comes since my early PhD studies in a way. And that's why I find macrophages quite such an interesting cell type, because they are very plastic. And we know from part of the work that I've just told you about that macrophages can directly play an important role in setting up this more fibrotic response after injury in the heart. But we also have data that shows us very clearly that macrophages are also important in fact, helping the heart to regenerate and to help new tissue to be formed. So if we think about it, we have one cell type. Of course, these cells are incredibly heterogeneous, so I'm kind of simplifying the system here. But you have one cell type that can do fibrosis or can do regeneration. And these two outcomes are quite at the opposite end of the spectrum. So what I'm really trying to understand now in my own lab is really how this programming is being set up. So when these macrophages go into the heart after injury and they populate the injured heart, they will be in contact with all the different cell types that make up the heart. So the heart is incredibly varied in terms of cells that make up the heart. You have cardiac muscle cells, you have blood vessels, you have epicardial cells, fibroblasts, and many different cell types. And therefore, depending where these macrophages are and which neighbors they're kind of more inclined to talk to, that will influence the way they see themselves in a way. So the way these programs are set up at the intracellular level. And what I really want to do, and what my team is now really working towards, is understanding how these programs are set up at the macrophage level. Because if we understand how the programming of a more pro-fibrotic macrophage or a more pro-regenerative macrophage is being done, that will give us much more knowledge to move forward and really ultimately be able to program these macrophages to favor a regenerative outcome over a fibrotic outcome. So this, in general terms, is what we are set to do.
Well, thank you so much for sharing. We'll definitely keep an eye out for your future research from your laboratory. So to end off our interview, I'd like to ask, do you have any central pieces of advice for any early career aspiring researchers?
Right. So I find that question always a bit tricky, because some advice that will work for me won't necessarily work for others. And there's always also a bit of a survival bias when you're giving advice, which I don't think it's really nice. but I guess by saying this is already maybe some kind of advice, which I would say, do listen to people that you highly respect scientifically, but at the end of the day, be true to yourself and do what makes sense to you, given your background, given your aspirations, given your own reality, and not necessarily what worked for A, B, or C. So definitely it is a bit of a challenge, but I think it's important that there's not one way, only one way to do what you want to do. But I can say, I guess, what's been quite important for me to in my career path to get me where I am today, and certainly is my passion about what I do. And I don't think I could be as committed or engaged if I didn't really care about the research question that I'm addressing, or if I would be forced to do something that really doesn't excite me much. But equally, for me, it's been the people that I've been exposed to. So for me, having peers, senior or junior, or it doesn't really matter, that are enthusiastic and creative and really spark some new ways of thinking about problems, that's really what makes it so unique and so spectacular to be doing science these days. So I guess, find your crowd if you can, and be always in an environment, either locally or, these days we can reach out to people all the time that can really, make you be a better scientist. I think that's something that, for me, I can truly say that's something that really impacts the way I do my science.
Mm-hmm. Thank you so much for sharing your insightful experience and research with us, Dr. Simões.
No, thank you. It's been a pleasure. Thank you so much for having me. And if I can just say, congratulate to you and your colleagues for such a great initiative. It takes a lot of determination as an undergrad to do this. So thank you so much for your time.
Oh, no, thank you. We really appreciate your presence on our channel. And thank you to all BEaTS Research Radio listeners for tuning in. Don't forget to like and subscribe to our YouTube, Twitter, and Instagram channels at BEaTS Research. BEaTS Research Radio is supported by the University of Ottawa Heart Institute, BEaTS Research Laboratory, and the Department of Microbiology, Biochemistry, and Immunology at the University of Ottawa. On behalf of our entire radio team and director, Dr. Emilio Alarcon, we hope to see you all next week. Thank you.